are ready to now have a look at our second um, dot point. So this is where we are going to be talking through lots of processes, and I do want you to um, yeah, feel comfortable as we go through them and understand that these will take a bit of time to wrap around your head. So, and I'll talk through some like study tips that worked for me when I first learned it, because definitely took a while to actually wrap my head around all the complexities of it. So the second dot point now is asking us to look at and consider how important is it for genetic material to be replicated exactly, um, and so we'll talk about that. Let's firstly have a look at meiosis and mitosis. So, mitosis is, as you can see here, this is mitosis. Now, before we do that, before we actually look at these two processes, we have to understand this concept here, to understand what is happening over here. So, this is a single chromosome. That's a single chromosome. Now, a chromosome, the way you actually count one chromosome is by the centromere. So, when a chromosome has one centromere, it's a chromosome. Um, so it's a chromatid essentially with one centromere, so it's a chromosome. And these are two homologous chromosomes. So homologous chromosomes are basically chromosomes that share similarities, like in terms of like things that they code for, where they code for, like in terms of height and size, they match up. So these are two homologous chromosomes. After DNA replication occurs, um, we end up with again. Now we're gonna end up with two sister chromatids, but it's still going to be one chromosome, but it's kind of going to be an X now. This is what happens after replication. So essentially, your body replicates this chromosome so that you've got this other attached to it, and the same thing happens on this side. But remember, I told you that it's counted by the centromeres. That's why it's still called one chromosome, even though now you've got two chromatids. You've got two chromatids, but it's still one chromosome because we count it through the center here, through the centromere. So this is what we end up with after replication, as you'll see here. That's the key that we end up with. So let's have a look. Now we'll start with mitosis. Now mitosis is done when the cells that when your body cells essentially need to be replicated. So when we need to form new cells within the body, the DNA will replicate the DNA, and it will do those processes. So that is where mitosis comes in. Um, there's a really funny, oh, actually, it's not even funny, but it's a joke that I was once told in class when we were learning. And it's like, what's one way to remember these processes? Well, my sister dropped something on my toe, and I said, "My toe, sis." I don't know if you laughed at that. It was kind of funny, sort of funny. I don't know. That I just remember it, so I don't know if that joke helps you remember the fact that it's mitosis. I think mitosis is the one that is done for body cells. Um, your body cells are known as somatic cells. So if you wanted, you can remember somatic and mitosis, like the T and the T in both of that. I'm not sure if that's how your brain works, if you remember things differently, but that's how mine works. I just like to make those little connections, and it just helps me remember. But sometimes I definitely do forget what connection I made as well, so that's totally okay. So here we are. We've got these are the processes. So let me write mitosis here. So this is mitosis. This is what happens. Alrighty. So we start off. The first process is the interface. So we start off with the interface. At the interface, this is where essentially this is where we are starting off with the chromosomes that in the nucleus. It's wrapped like obviously it's, you know, within the formation of the cell, and we also have these centromeres starting to come through as well. Then in prophase, essentially what happens is now these spindle fibers, they start to extend. Can you see here? So in interphase, the, you know, the cell's getting ready. So we've got these centrosomes here, and now we've got these centrosomes and the spindle fibers ready to go. In prophase, the spindle fibers, they start to extend, right? Think of them as like spidery extensions. They start to like expand, and so they expand, and then we've got metaphase, where now, and between that is prophase, pro-metaphase, when these chromosomes line up. But metaphase, they've all lined up now. You've got the homologous chromosomes lining up this way, and the spindle fibers have attached to the chromosomes in each side, as you can see here. So it's attached on each side. And then anaphase is when they've attached and now we pull them apart. And then in telophase, we pull it apart so that we will now have these the same number of chromosomes. We'll have 46 because this is a body cell, so we'll have 46 in both of them. So that's happened here now, telophase, and now slowly the cytoplasmic wall comes in, and then cytokinesis finishes it off, and you get two new cells here. So this is basically how new cells in your body are made. So let's go through that again. This is how new cells in your body are made. We started with one cell, right? The one cell has the DNA in it. It's an interphase, and then it goes into prophase, where now the spindle fibers start to will start working. They've opened up for the morning, so they start to work, and then we see these homologous chromosomes line up. And then once they line up, in the spindle fibers attach on each side, and then they pull it apart. Once they pull it apart, and then we see the nuclear sort of capsule sort of pack around it. So the nucleus sort of comes around the DNA, around the chromosomes again, and obviously the chromosomes have our DNA within them. And once that is done, cytokinesis happen, and you're left with two cells. And this is what happens when your body is making new cells. If it's getting rid of old cells, you know, or you've hurt yourself, or you have hurt yourself, and it's making new cells. All your body cells will have the same DNA, and they'll have those 46 chromosomes that make you you. Now that's mitosis. Alrighty, let us now have a look at meiosis. Now meiosis is interesting because it differs from mitosis in the number of rounds. So the rounds of division. So meiosis happens in 2 rounds. There's meiosis 1 and meiosis 2. It also differs in the number of daughter cells that we end up with. The chromosome numbers also differ in the cells, because remember, it's meiosis now. This is where the gametes are being made. Alright, and now because the gametes are being made here, what we will see is that it's not going to have 46 chromosomes. It's going to instead have, hopefully you were listening, it's going to instead have 23 chromosomes instead of 46. Alrighty, so here we are, and the role in the body is different. These are your normal cells. They're going to go back and obviously help with the body. Your gametes are for sexual reproduction. They are going to be utilized when fertilization is about to happen. So that's when they're going to be utilized. So let's have a look at how meiosis works. So meiosis, we start off with interphase. Same way, we've got our chromosomes ready. There's still a nuclear envelope around our chromosomes. The mitotic spindle is sort of here, and this way we've got our centrosomes as well. Then DNA is copied, and once DNA is copied, prophase starts. So I think one thing I've forgot to mention here with interphase is that DNA is copied. That's really important. So what happens is, in interphase, DNA has been copied for it to go. So without DNA being copied, the process can't start. And let me show you something. And now I do not have a pen with me, so I'm going to draw using my curse pad. So please Please go easy on me. Let's have a look. So this is how the chromosome starts. Alrighty. In interphase, this chromosome is gonna, so this is the chromosome. Alright, bear with me. This is the chromosome, that's the centromere in the middle. This is the chromosome. So here we go. There we go. Alright, that's a chromosome. Now, when DNA replication happens, remember what happens if we've got this one chromosome here, like this? It becomes 2. It has been copied. That's what the second thing is. It's a copy of the first one. This one here is a copy of the first one. So it has been copied now. And that is why here we see like the X. That's why it comes apart, because they've been copied. That is what is happening. Alrighty, so I forgot to mention this here in interphase, but in interphase your DNA is copied. This is where DNA replication occurs, because obviously if the DNA is not copied, what are we going to put into the new cell? Like, we can't have a cell without genetic material, because everything that the cell does, it takes instructions from the DNA. So we can't obviously have a cell without genetic material. That does not work. So we actually first copy the material in this in our existing cell. We copy it. We end up with 2 chromosomes, and then we put them apart. And so you've still got 46 chromosomes in each, but you've got them like this now. Alrighty. So let's move on to meiosis now. So same thing. DNA is copied. The chromosomes pair up. Now, in meiosis, a very interesting process called recombination occurs. So and it's called crossing over. So what happens is the so they've lined up. They've got now the mitotic spindle has attached to them. In metaphase 1, the mitotic spindle has attached to the 2 chromosomes. But before they're pulled apart, they're going to cross over. They're going to cross information over. They're going to exchange info. Think of it like they're going to exchange numbers so that, you know, I don't know, they can keep in touch. I don't know. But they're going to exchange numbers. So once they do that, you can see here in anaphase 1, they're pulled apart. In anaphase 1, they're pulled apart. And so then telophase and cytokinesis happens, cells pinched in the center, and then it breaks apart through cytokinesis. Alright, so now in prophase, what happens is we've got the 2 daughter cells. These are called the 2 daughter cells, so they line up. The 2 daughter cells line up. Now the independent assortment does not occur, so that's called prophase 2. So the naming is the same, but it's just that when it's a second cycle, it's like you write a 2 in front of it. So here we are, prophase 2, they're there, and then in metaphase 2, the mitotic spindle attaches to them, and now they're going to be pulled apart. So they're going to line up at the equator, at the center of the nucleus, and then they're going to be pulled apart in anaphase 2. They're pulled apart, and then in telophase 2 and cytokinesis, we end up with 4 daughter cells. But the thing here is now, each one of these daughter cells, they're going to only have 23 chromosomes. Only 23 chromosomes, not 46 chromosomes. Here we had 46. Here they're only going to have 23 chromosomes. So as you can see here, this is a very simplified diagram which shows you just 1 chromosome there. There's only 23 though in each of them. So really important here is this process of crossing over. So what happens is when we start off with interphase, and I might just pull up a screen. Okay, so when we start off in interphase, we start off like this, right? This is my chromosome. I'm going to try and make a circle there. It's a big circle for a chromosome. And this is my chromosome. Alright, so we're going to start off with these chromosomes here, and then we're going to copy them. We are going to, where is it? Okay, we're going to replicate them so that we can have the DNA material. So that's chromosome 1, that's chromosome 2. Alright, now these two, they are going to line up, and this is going to happen to all the chromosomes. They're going to line up. Here we go. Going to line up here. Now, before anaphase, before they're pulled apart, what happens is when they line up, they exchange information. So let's say, and I will make this a bit different now. So let's say that, okay, this is this chromosome. This is how, no, let's say this is this. There we go. So let's pretend that my second chromosome here is green and the first one's blue. Alright guys, so there we are. So now, after they've lined up, they have exchanged genetic information. Because they've lined up homologously, that means that they code for this, for similar areas. That's what it means. So let's say they've crossed over alleles, they've crossed over the genetic material for hair color. And that is how, essentially, diverse, that's how basically you see like kids having different traits to their parents. Like I know someone who does not have like red hair or blue eyes. Their partner doesn't have red hair or blue eyes, but their daughter has beautiful red hair and blue eyes. So it's because it skipped a generation. So that's how these alleles work, because of this crossing over, this independent assortment. So here we are, and this is what's happened now. So we've exchanged these alleles, and so now there's a new combination of alleles basically, and that's diversity right there in the process. That is diversity. Alrighty, so hopefully that makes sense. There we go. Oh, look at me writing diversity all the way through with my curse pad. Amazing. Okay, so this is essentially what. Oh, where did it go? Okay, there we go. So that's essentially what's happening in that process there. So key thing is mitosis, you end up with 46 chromosomes in the cell. Meiosis, you only end up with 23 chromosomes in the cells. So this was, let's say this a female cell's undergoing meiosis, these are going to be the eggs, right? These are going to be the eggs, and the eggs are going to have 23 chromosomes each, but they're each going to have obviously different, they're all different from one another. These are not similar to the parent organism, and they're not similar to each other. So really important that you understand that. And I would say, honestly, hopefully this helped. I really hope this helped. Now, if you have any questions, I hope you've been asked. I hope you have been asking, but I think the best way to actually get a really good understanding of this idea further is to watch YouTube videos, like the ones with the animation. They really help, like with the visual understanding. I mean, I can try, yeah, I tried to draw for you, like we clearly saw how good my drawing was. No, but honestly, YouTube videos are really good. And the one that I really like, the channel is Amoeba Sisters. So if you search up Amoeba Sisters, they are really good because they're like images, like the drawings are really nice, and they explain it really nicely as well. The animation makes it so much more easier to understand some of these processes. So definitely recommend checking that out. But yeah, look, this is not a easy process to wrap your head around, so I would recommend that you definitely check out those like other resources to get a really good understanding. And drawing diagrams can always help, but hopefully today is a good start